Hi everyone. Today we begin our lesson with chapter number 5, enzymes. In this chapter we are going to explain by enzyme as biological catalyst and properties of enzymes. We'll also explain key definitions. Number 1, catalyst. A substance that increases the rate of chemical reaction and is not changed by the chemical reaction. Catalyst may be inside the living organism or it may be outside the living organism in industries. Enzymes are proteins, they function as biological catalysts, means they speed up chemical reaction inside the bodies of living organisms. What biological catalysts are? In the bodies of plants and animals, hundreds and thousands of chemical reactions are taking place all the time. Collectively, all these chemical reactions are called metabolism. Metabolism is actually a two-step process in which smaller molecules link up with one another to form larger molecules. This is said to be anabolism, and another step is catabolism, in which larger molecules are broken down into smaller ones. That is catabolism. So anabolism and catabolism are collectively known as metabolism. For example, digestion. Digestion is an example of catabolism because larger molecules of food are broken into smaller and simpler ones. Okay, same is the case with the photosynthesis. Photosynthesis is example of anabolism because small molecules like carbon dioxide gas and water link up with one another to form larger molecules like glucose. Almost all of these chemical reactions are controlled by enzymes. Enzymes increase the speed of chemical reaction, hence they are biological catalysts. We are going to explain digestion as an example. Definition first. Large molecules of food are broken down into smaller, simpler and diffusible form with the help of enzyme. This process is said to be digestion. Digestion takes place in the alimentary canal which starts from the mouth and ends at the anus. This whole tube is collectively known as alimentary canal and the process of digestion takes place in here. Just because the shape and function of different parts of the alimentary canal is different, therefore their names are different. We can digest only protein, fats and carbohydrates. All these three are giant molecules. Now there are actually 7 categories of nutrients. Or the other 4 are water, fibers, vitamins and minerals. But water, fiber, vitamins and minerals cannot be digested. For example, fiber. Fiber are the undigested part of the food. We do not have enzyme to digest these substances. Water, minerals and vitamins, these are already in simplest form. They do not need any further digestion. So the only 3 molecules or the only 3 components that can be digested are protein, fats and carbohydrates. These are giant molecules and they cannot pass through the wall of the small intestine into the blood. So they first must be broken down into their specific units. Carbohydrate, proteins and fats are digested in intestine where they are broken down into their basic units. Carbohydrates into sugar, proteins into amino acid, fats into fatty acid and glycerol. Enzymes are also found in plants. Reactions like photosynthesis and respiration in plants are controlled by enzymes. Remember one thing, photosynthesis and reactions are chemical reactions and they are controlled by enzymes like other. Photosynthesis is a chemical reaction in which carbon dioxide reacts with water to produce glucose. Same is the case with respiration. It is a chemical reaction in which food is oxidized to release energy. Both of these are enzyme controlled chemical reactions. We are going to explain 2 of the enzymes. Number 1, catalyze and number 2, starch phosphorylase. Catalyst first. During the chemical reactions, one of the byproduct called hydrogen peroxide is produced inside the bodies of plants and animals. Hydrogen peroxide is dangerous and must be broken down immediately. So catalase is an enzyme. They work inside the cell and break hydrogen peroxide into water and oxygen. Water and oxygen are not dangerous to the cells. Starch phosphorylase breaks starch molecule from glucose inside the plant cells. Now, how to name enzymes? It's very simple. Enzymes are named according to the reaction they catalyze. For example, enzymes which catalyze the breakdown of carbohydrates are called carbohydrates. If they break down proteins, they are protease. If they break down fats, they are lipase. Sometimes they are given more specific name than this. For example, amylase break down starch, maltase break down maltose, and sucrase break down sucrose. The lock and key mechanism. An enzyme which is a lock work by allowing the molecule of the substrate key on which it is acting to fit into it. The fit has to be perfect. We say that the shape of the enzyme and the shape of the substrate are complementary to one another. Look at here, number one. A. This is the lock enzyme and this is the key substrate molecule. Now, the shape of the key must be complementary to the lock, so the lock opens. Now, this is actually the shape. Now, we turn the key and the lock opens. Same is the case with the enzymes. This is enzyme. The active site of enzyme must be complementary to the substrate. Okay, the shape of the substrate exactly fits into the enzymes. Now, the molecule, this is said to be enzyme substrate complex. Now, look at here this example. This is enzyme, the gray color, okay, and these are actually the substrate molecule. The shape of the substrate molecule must be complementary to the active site of enzymes. Okay, now as you can see, these molecules can exactly fit into the active site of the enzymes. The enzyme substrate complex is produced and when product is released. Now this enzyme can be used for over and over again. Enzymes are not used in the chemical reaction and they can be recovered at the end of the reaction. Now we are going to explain one of the very important topic that is called active site. What is active site? The active site of enzyme is specific region or pockets on the enzyme surface where the catalytic activity takes place. It is crucial for enzyme's function as it facilitates the binding of the enzyme to its substrate and catalyzes the conversion of substrates into products. Look at here, we have got an enzyme and we have got two substrate molecules. The substrate molecule exactly fit into the active site of the enzymes. Now you can see that they fed into the enzymes and the product is formed. This structure is now said to be enzyme substrate complex and the product is released. This enzyme can be used for over and over again. Okay, now a single enzyme is capable to to, to convert hundreds of substrate molecule into products. Properties of enzymes. All enzymes are proteins. They may seem odd because some enzymes actually digest proteins. Enzymes are made inactive by high temperatures. This is because they are protein molecules which are damaged by heat or high temperatures. Enzymes work best at the particular temperature. Each and every enzyme has an optimum temperature at which they work best. For example, enzyme in a human body work best at 37 degrees centigrade. Below this temperature or when the temperature gets higher, the activity of the enzymes are compromised and sometimes the enzyme become denatured, means unable to catalyze any more reactions. So optimum temperature for enzymes in a human body is 37 degrees centigrade. Enzymes work best at particular pH. pH is the measure of how acidic or alkaline a solution is. Some enzymes work best in acidic solution, low pH. Other work best in neutral or alkaline condition, high pH. Look at here. For example, in a human body, the enzyme they work inside the human body work best at 37 degrees centigrade. For example, this is actually the peak, the high performance of the enzymes. Okay, now you can see that if we drop down right here, so this that might be like 37 degrees centigrade. So a from 0 up to 20, 30, etc. As we increase the temperature, the rate of the reaction increases until 37 degrees centigrade. As we increase the temperature like 40, 50 or more, so what happens? The activity of enzyme drops. So at a certain temperature like 37 degrees centigrade, the rate of the temperature, the rate of the reaction is highest. Same is the case with pH. Most of the chemical reaction was work best at pH 7. Some ate acidic or other ate alkaline. Okay. For example, this is actually the optimum pH at which enzyme work best. Enzymes are catalysts. They are not changed or used up in the chemical reaction which they control. They can be used over and over again without being consumed. A small amount of enzyme can change a lot of substrate into products. Enzymes are specific. Enzymes are highly specific in their actions. The active site provides a unique three-dimensional microenvironment. They measure the shape and chemical properties of the substrate. This allows the enzyme to bind specifically to a substrate while excluding other molecules that do not fit precisely into the active site. For example, an enzyme that are made for carbohydrates cannot work for proteins, and an enzyme that are made or are complementary to the protein will never work for fats molecule. Means they are specific. They can work only one type of substrate molecule. Temperature and enzyme activity. Low temperature slows down enzyme activity. The optimum temperature maximizes enzyme activity. High temperature can denature the enzyme, causing a loss of activity. Maintaining the proper temperature is crucial for ensuring optimal enzyme activity in biological system. Look at here. For example, 37 degrees centigrade. In humans, this is actually 37 degrees centigrade or optimal temperature for most of the chemical reaction inside the human body. Okay. Now you can see that this is the peak performance of the enzyme at 37 degrees centigrade. As we increases from 0 to 10 to 30, the rate of the reaction increases until 37 degrees centigrade. As we pass 37 to 38, 39, 40, 50, 60, and the rate of reaction drops down. Okay, because high temperature denature enzyme, the enzyme may not be able to accommodate substrate molecule any further. pH and enzyme activity. The pH of a solution affects the shape of an enzyme. Most enzymes are their correct shape at a pH of 7, that is neutral. If the pH become very acidic or very alkaline, then they are denatured. Denature means the enzymes, the shape of the active site of an enzyme change. Now the enzyme cannot accommodate or are not complementary to the substrate molecule. This means that the active site is no longer fit the substrate, so the enzyme can no longer catalyze its reactions. Some enzymes have an optimum temperature that is not neutral. For example, protease enzyme in stomach that has an optimum temperature about 2 degrees Celsius, 2 pH, sorry. Increasing enzyme activity. Now, this is slightly alkaline, okay, optimum pH level. So, we is from, let's suppose this is 4.5, 5, 6, 7, 8. Now, at 8 pH, the rate of the activity is higher. As we move from 8 to 9, 10, and 14, the rate 14, the activity of the enzyme decreases. So, at optimum pH, the activity of the enzyme is highest.